Nature Center for our monthly nature seminar. This month I'm thrilled to have Hank Gruner here with us. He is Vice President at the Connecticut Science Center in Hartford, also a leader of the Connecticut Amphibian Monitoring Project. And he's recently done some research on salamanders in Wintergreen Woods, the nature preserve here in town. We've been chatting about this for a while, and I'm thrilled to hear a little bit more about his current research. Thank you so much for coming and spending time with us.
Glad to.
Thanks.
Have to come out, and thank you all for coming out tonight. Just gonna clip this thing on here now. Hopefully. Good?
Yep.
You all right? Perfect.
Yeah.
Okay, great. I'm gonna stand over here so I don't block your view. How's that? I don't block poor Chris's view.
And whenever you like, if you can hit that light. Or you or
Yeah, see what
you need.
how we do. That good?
That's better.
All right, you like that better? All right, good. All right, let me give you a little my background first, and then I'll get into this story here, because it actually is a really true mystery about, well, we're looking at certain salamanders that you're going to see, and it took us about 4 years to solve them. I'm happy to say that this year, I called Chris earlier, I think it was, we were getting on last spring, and I said, geez, we want to collect some additional salamanders, we're trying to solve this mystery. And he said, sure. So we went out there last spring, and we were able to collect a sample that solved the mystery finally after about 5 years of study. And we'll go through that mystery a bit. But my background is, as Chris mentioned, I'm the Vice President of Programs over at the Connecticut Science Center. And my background is in conservation biology. So I do a lot of work working with the state of Connecticut and with developers and with nonprofit organizations, primarily on the conservation of amphibians and reptiles. That's my area of specialty, of which the salamanders are one of the amphibians that we have in Connecticut, along with frogs and toads. But I also do work with various reptiles, snakes and turtles in the state. And as a group, especially amphibians, have a lot of conservation challenges. We're seeing a lot of declines throughout the world in amphibians. So a lot of the work that I do is designed to provide information to help us conserve the species of salamanders and other reptiles and amphibians here in Connecticut. And I've done that work for about 30 years. Even though I work over at the Connecticut Science Center doing a lot of educational programming, I still continue to do a lot of this work with colleagues. And in fact, many of the slides you're going to see, I'm not really a great photographer, so many of the slides you're going to see are not mine. I borrowed many of them from Dr. James Bogart, who is out of Guelph, Canada, at the university there, where he is the individual who did a lot of the genetic analysis of the salamanders that we collected as part of a project that I worked with Dr. Michael Clemens on, who for many years worked in Connecticut, published a book called Amphibians and Reptiles of Connecticut in 1993. We're actually redoing that book now. We're in the process of writing that book and redoing the maps to look at the picture 20 years later. So Mike was working on that, and then I also borrowed some images from a group of Trinity students who did some work looking at the wetlands in Wintergreen Woods. So I wanted to make sure I pass on the credit for some of the good photographs that you're going to see to James and the Trinity students. So let me tell you a little bit about the mystery at Wintergreen Woods, and it involves that critter right there. So let me set the scene. It's a true mystery, so we're going to set the scene. The scene is Wintergreen Woods, and the focus that we want to look at in Wintergreen Woods, because there's some nice forest land in there, but we're very interested in these critters. Now this picture is not from Wintergreen Woods. This is actually a vernal pool in Massachusetts. Has anybody heard of a vernal pool? You ever heard that term? A vernal pool is a term for a wetland. It's in vernal refers to spring, which we tend to see where it fills with water generally in the spring due to snowmelt and heavy rains. They tend to be fairly small in size, very shallow, and they do something that most wetlands don't do. That's the same site. Where did the water go? It dried up. So vernal pools, or seasonal wetlands as we call them, don't hold water year-round. So basically they dry up. You tend to see them in the spring, and then they'll hold water until July, August. It depends on the pool, it depends on the year and the amount of snowmelt and the amount of rain that we have. And then they tend to dry up. Sometimes they'll fill in the fall rather than the spring, but generally they have to dry up. Because they do that, they don't have fish in them. Fish can't colonize them. So in the absence of fish, many species of amphibians have adapted to use them primarily to breed. The amphibians don't live in the water year-round. They're actually forest animals. They live in the forest, and they come to those vernal pools usually in the spring. We have one species that does it in the fall. And almost now, in fact, I'd be willing to bet last night, was it last night that we had the heavy rain and it was a little warm? I'll bet last night some of these guys already started moving into that pool, the pools that over here in Wintergreen Woods. They're very cold tolerant. So what they do is they breed, they lay their eggs in those pools. Their young develop very quickly in a period of months, and then they leave those pools. So the animals, the mature animals, the adult salamanders, actually live out in that forest. And they only return to those pools each year basically for a short period of time to lay their eggs, to reproduce, and then to leave. So they're a unique type of wetland or watercourse that is found primarily throughout New England because of the glaciers that we had formed many of these. But if you go out to California, you can find interesting vernal pools. If you go to the southern United States, they'll often have the names precocins. They're called by different names often depending on where you are, but they have the same functions. And the reason they're different than many of the other wetlands like lakes and ponds is because they don't have fish. And fish are a predator. So if you were to introduce fish, they would eat all the salamanders. Those salamanders are not found in other places besides these vernal pools. So that's the scene. It's wintergreen woods, and our attention is really focused on those vernal pools. This is a map from the students from Trinity. And Chris, do you remember what year they did that study? I don't remember the year that they had. It was in the late 90s, wasn't it? Early 2000s, something like that?
I thought it was more recent than that.
Oh, could be, yeah. So this is an aerial photograph of wintergreen woods, and all the areas that are outlined in green there and colored in that orangish coloration are basically areas where there's those type of vernal pools. We often call them pingo potholes in this region, or kettle pools. They tend to be, and the difference is that they tend to be very circular. Some of those can be a little deeper, and they formed with basically blocks of glacial ice. So you'll also often see large clusters like this. I can take you to, I did a project where we mapped vernal pools in 7 towns in the Connecticut River Valley. One of those towns was Suffield, it was right within the river valley, and then the others were more associated with the Farmington River and the uplands. And we mapped 1,300 vernal pools. 700 of those vernal pools were in the town of Suffield. The other 6 towns split the rest. And the reason was because Suffield is very similar to the landscape here in Wethersfield. It has that same soil composition. It was part of that glacial lake plain with Glacial Lake Hitchcock. So you get clusters in areas of these potholes or kettles or vernal pools. So that's looking down on wintergreen woods. And those are the cluster of areas where we were very interested in. All those vernal pools and what they might be holding. This is an image from wintergreen woods. So there's one that you can see surrounded by the forest land. You can see a little ice on that still, so that's either in very early spring or fall, late fall, somewhere there. So now let's look at the suspects. We've set the scene for our mystery. The scene is wintergreen woods and the vernal pools in there. And the suspects are what we call a group of salamanders. In Connecticut, we have 14 different types or species of salamanders. There's a family of them, a group of them that we call the mole salamanders. And there's a reason. They spend most of their time as adults underground. You tend not to see them. You can find them occasionally if you lift up rocks and logs, but they tend to spend most of their life underground, except in the spring when they come out to breed. Their genus, when we look at them scientifically, they're called Ambystoma. That's what we call them. They're Ambystoma salamanders. And let me show you the cast of them. These are the ones that are found in Connecticut. In other parts of the country, you would find different species, but these are the ones that are found throughout Connecticut. We have the Jefferson salamander on the left, and the blue-spotted salamander on the right. We have the spotted salamander on the lower left, and the marbled salamander on the lower right. The Jefferson salamander is a species of special concern in Connecticut. You're familiar with endangered and threatened species, right? Those are animals or plants that are not doing so well and could become extinct. In Connecticut, we have 3 designations. The state calls them either endangered, if they're not doing very well at all, generally less than 5 populations across the state. Threatened, which means generally less than 9 or 10 populations across the state. And then special concern, which means that they're not really widespread, but there's still a fair amount of populations in the state, but we see declines, we see evidence of problems. The Jefferson salamander is a special concern. The blue-spotted salamander is both special concern and endangered, which sounds confusing, but I'll explain it a little bit later. The spotted salamander is fairly common throughout Connecticut, where you have vernal pools and you have forests. It's our most common Ambystoma salamander. The marbled salamander, the little black and white guy there, is also not listed, but not as common. When you get into the high elevation areas of northwest or northeast Connecticut, you don't find them. But here in central Connecticut, you can find them, especially up along the ridges, Talcott Mountain and so forth. The odd thing about this guy is, these all breed in the spring, this guy breeds in the fall. They deposit their eggs in the fall in the dry bottom, then when the rains fill, their larvae hatch, and they grow big. What do you think those big larvae feed on in the spring? Babies of these guys, and also of frogs. So this guy's a little different. The marble salamander has a little bit of a different breeding cycle. I guess they thought it was a little too crowded in the spring, so they decided to spend some time in the fall. So that's the group that we're interested in. But let me show you what's happening at Wintergreen Woods. So what we're interested in is these two species, the Jefferson salamander and the blue-spotted salamander. And the reason we're interested in them is when we started, and this dates back to 1950s, 1960s, researchers studying them started to see something very unusual. When they were looking at a population of these salamanders in a vernal pool, they were finding very few males, and in some cases, no males. So we call those unisexual populations, which basically means there's only one sex. So how do they reproduce? Well, unisexual animals are not unknown. In fact, there's about 80 or 90 different species of organisms that reproduce unisexually. They basically can be like clones. There's different ways that they can do that. But with the salamanders, they started seeing some interesting patterns associated with the salamanders. When we got a little bit more sophisticated and started looking at the genetics, it gets really interesting. Here's what we have at Wintergreen Woods. The guy on the left is the blue-spotted salamander. That's what we have at Wintergreen Woods. This guy doesn't occur there, as far as we know. These two here are the hybrids. So that's a pure Jefferson salamander. That's a pure blue-spotted salamander. This guy over here is a hybrid between the two, but closer to blue-spotted salamander. You can sort of see that in the coloration and the looks. This is a hybrid, they're both females, and it's closer to the Jefferson salamanders. In Connecticut, in fact, when I started studying amphibians and reptiles in Connecticut, these were considered separate species. In fact, you can find some books that'll still call them. That's the Tremblay salamander, and that's the silvery salamander. We no longer recognize them as species, because as we understand the genetics, we understand they're related to these blue-spotted Jefferson, so we consider them what we would call a complex. These are always female. Never found the male of this complex down there. Only when they're pure do we find the males. So let me show you how bizarre it actually gets. When we started studying them throughout the northeastern United States, going from from Canada and Ohio, parts of Michigan, out through Pennsylvania, New York, up into Maine, as far south as New Jersey and in southern Pennsylvania, it got really interesting. We find that this is a tiger salamander. You already met the Jefferson salamander and you met the blue-spotted salamander, and then there's a smallmouth salamander here. We found that there are 5 or 6 different species that hybridize with one another. And in fact, in some ponds in Ohio, when we look at the genetics of one individual, it has the genetic DNA of 5 different species within it. Here in Connecticut, we only have blue-spotted and Jefferson salamanders, so we don't get tiger salamanders. We don't get the smallmouth salamanders, so we don't see that combination. We see combinations genetically between the blue-spotted and the Jefferson. But it gets even stranger. As we get more sophisticated with our genetics, there was another species that showed up, a species called streamside salamander. And what is really fascinating is these animals are behaving like no other vertebrate organism or backbone animal on Earth. What we have found is that these are actually very ancient animals that have 2 genetic structures to them. One of them is that they have a combination of those 5 species that I mentioned. Their mitochondrial DNA, the DNA that comes from the mother, can be a combination of Jefferson, blue-spotted, streamside salamander, any one of that group. But their nuclear DNA of all of them is more closely related to this species, the streamside salamander. and very different than the mitochondrial DNA. So what we believe has happened, and you'll see why it's a mystery here, what we believe has happened is that when we look at the range of these species, this is where these all occur, okay? You can see the colors. The reddish coloration is the Jefferson salamander. It's kind of hard to see. The blue spotted is the gray. The smallmouth salamander is the green. Streamside salamander is the darker blue. And the tiger salamander is the yellow. You can see sort of where they occur. But what we believe has happened is that about 5 million years ago, in the area of western Kentucky, there was an event, a hybridization event, that led to the development of the complex that you see today. And that ancient genetics is preserved through all that time in every one of these species, basically in their nuclear DNA, but they're doing some interesting things with the other Jeffersons and the blue spotted with the other species. So it's a complex genetic thing that's going on, which we're still figuring out as our ability to look at genetics gets better. So it involves all of these species, 6 different species of salamanders basically hybridizing. So where does wintergreen woods come in? I know that's a little complex. We can dispense with that. You might ask, how do they reproduce if you have lots of females? This is normal reproduction. We all understand that. You have a male and a female, and you can both contribute chromosomes. You can have what's called hybridogenesis, where basically a male and a female will breed, but you will get a complement of the male sperm, but it will be basically kicked out. You can have gynogenesis, and you can have parthenogenesis, which basically doesn't require a male at all. It's almost like cloning. If you're familiar with cloning, it's almost like cloning. Our salamanders don't do any of this. They do this, this, and this. And in fact, they do a new form of reproduction that's never been described before. We call it kleptogenesis. Does anybody know what a kleptomaniac is?
Someone who steals stuff?
Somebody who steals stuff. Kleptogenesis basically means that what we have found is that with the female salamanders, the unisexual salamanders, they only can persist by stealing the sperm from any one of those other males. So you saw those other species, the Jefferson, blue-spotted, tiger salamander, streamside salamander, smallmouth. The only way that those unisexual salamanders persist is basically by stealing the sperm or reproducing with one of those wherever they occur. So if they're, in our case, it has to be from a blue-spotted or a Jefferson salamander male. That's where they get their sperm. It may be incorporated in so that that genetic information is passed off, or it may not. It varies within the populations, but they require, the unisexual females require, those hybrids require those males to basically help produce. So we're getting to the mystery of wintergreen woods. What we also see when we look at, these are actual chromosomes. Does anybody know how many chromosomes we have? Any guesses?
19.
We have actually 23 pair of chromosomes. So a total of 46. We have 23 pair. These guys, what we tend to see is when they reproduce, they'll have elevated numbers of chromosomes. That's a normal. So that would be like a Jefferson salamander that's not a hybrid or a blue spotted. And you can see it has 14 pair, basically 28. Here we see when they hybridize, you'll get an elevation in the number of chromosomes. And we can actually see that. We can use microscopes and we can see that. So we would call this a triploid. We would call that a quadruploid. You could have up to a pentaploid. You find all of these complexes of chromosomes in these salamanders. And that's how we used to identify them, by the way, was counting those chromosomes. Now, you get a little bit more sophisticated, you can actually stain the chromosomes. The green ones are from the blue-spotted salamander. The red ones are from Jefferson salamanders. So we can start to really look at it with techniques where you can use fluorescent dyes and really look at the different chromosomes and really get a sense of what's happening. So our understanding of genetics, by the way, not just for salamanders, but for people in genomics, is really a revolutionary area, field of study. There's a lot going on. Here in Connecticut, by the way, We're one of the leading states in genomics research, especially for people. I don't know if you've seen the Jackson Labs, which is moving into the state, the University of Connecticut, Yale University, Wesleyan University. There's a ton of top-end genomics research going on, a lot of it targeted at human disease. And I guarantee you, we're working on a genomics exhibit at the Connecticut Science Center. I can tell you just by looking at all the information that's out there, this is an area that's rapidly growing, and there will be a lot more breakthroughs that you'll see in the coming years because there's a big focus in this area. And a lot of that's happening right here in Connecticut, and will continue to grow in Connecticut. But we also use it in wildlife, so you can see some of the techniques. When I was in a high school, you couldn't do any of this stuff. And in fact, when I first started, when I got out of college and was working at then what's now the Children's Museum, we used to use a technique called electrophoresis, where you pass electric current through some samples and you get these what are called protein bands, and you can look at the bands. That was cutting edge. That was state of the art, what universities like Dr. James Bogart were doing to analyze these. High school kids today have been doing electrophoresis like that for probably 5 years, at least 5 years, I'd say, maybe more. In high school classes, they've been using that technique. DNA sequencers, what used to take $30,000 to sequence a genome, now costs about $1,000 or $1,200 to sequence a genome. So it's a complete genome, not just a segment, the entire genome. So genetic technology is really helping us understand what's happening, not only with these creatures, but with ourselves. So let's look at Connecticut. We're going to narrow down to wintergreen woods in a few. Let me show you what's happening in Connecticut. The green dots are basically Jefferson salamanders that are pure. These are not, make sure I got my dots right. The red dots, I'm sorry. The red dots are the Jefferson salamander complex. The blue dots are the blue-spotted salamander complex, which means they're all hybrids. So when you see a red and a blue dot, you see they're all hybrids, okay? When you see the green triangles or the green over here, those are pure. They're not hybrids. You're seeing pures. Remember when I said that the blue-spotted salamander had 2 designations? It was endangered as well as special concern in Connecticut. Here's why. This population here, which used to be in Rhode Island, no longer occurs there. But this population here is pure, genetically pure. We've never found hybrids associated with it. They have not mixed at all. All the blue-spotted salamanders, like in here in Wethersfield area, are all hybrids all down this area. So we recognize in Connecticut, very progressively, genetic differentiation. So we're saying that the blue-spotted salamanders down here are very vulnerable, they're genetically distinct, they're considered endangered species. Versus the ones that are hybrids, they're more widespread, not as vulnerable. We consider them to be basically species of special concern. There's a reason for this, and it has to do with glaciers, basically. That's what we believe. That at one time we were covered by up to a mile sheet of ice. Glaciers moved through this area several times. The last time was 10, 12,000 years ago, Wisconsin glaciation. And what happened, obviously, is as this was covered with glaciers, you didn't have the forested habitat. Any organisms that are there were either wiped out or had to gradually move off because the glacier it's not moving fast. That obviously happens over time. So they pushed off. Long Island Sound wasn't there. This was actually land out here. Georges Bank was above water. That's where they go fishing now. That wasn't then. A lot of the water was bound up in ice. We believe what happened is the ancestors of these species sort of moved south to avoid the glacier over time. Some went this way, some went this way, down south or off the Georges Bank. As the glaciers receded and organisms, both plants and animals, recolonized New England, you had certain things. You had a big lake here called Glacial Lake Hitchcock, which may have served as a barrier for these guys to get over in here and hybridize. So they never had a chance. These guys really never came back in contact. So they're separate. They're genetically separate. So in Connecticut, throughout New England, a lot of what you see with our habitats and the distribution of our plants and animals is associated with what happened with glaciers and the recolonization of our state by plants and animals. And there's been studies done that have tracked how trees have recolonized and the progression of that, and how different organisms have recolonized fish into the streams and things like that. So a lot of what we look at is associated with what happened with glaciers over periods of tens of thousands of years. So that's what the picture looks like a little closer here in New England and Connecticut. So let's look at what's the mystery here. So the mystery in wintergreen woods is this. In 2000, we first found the blue-spotted salamanders in wintergreen woods. I found 5 of them, juveniles, very young ones, in a dirt pile next to a parking lot. And I was doing a study. I was very interested in looking at urban parks. I was curious as to what species can still survive in urban parks. And I was looking at Keeney Park in Hartford, which is over 800 acres. I was looking at, I think it was 30 different parks throughout Connecticut. I had seen wintergreen woods, said, wow, this is a small forested area. We'll check that out. So we found, in 2000, we found the first sample of these. And we were still collecting samples throughout the state for the studies. You saw the dots for those studies, doing genetic analysis. James Bogart was doing all the work. Dr. Michael Clemens, and he had published a lot of studies on this. So I submitted them in for analysis. And Dr. Clemens contacted me the following, after we submitted those, he said, Hank, you got an interesting group of salamanders there in Folly Brook over in Wintergreen Woods. I said, what's up with them? And he said, well, they're all, what we call in the business is, they're all LJs. Well, an LJ simply means it's got 1 complement of chromosomes of blue-spotted salamander and 1 complement of Jefferson's. It's a hybrid that we would call a diploid hybrid. In other words, doesn't have extra chromosomes. And they're all females. So there's something going on between Jefferson's and blue-spotteds, but there were no males. They were all females. And they were all the same genetic group. Generally, we would see some that would be LLJ, or LJJ, or LJJJ. We'd see different mixes. Occasionally, we'll get these, just like the LJ, but usually not all of them. So we went back in 2001. We got some more salamanders. Said, well, we'll figure it out. It's a low sample size. We got 5 salamanders. We'll figure it out. So we got some more salamanders and sent them back. And it takes a few months to do all this because everybody's busy. So I get a call back the following winter. It's like, Hank, we still got all LJs and they're all females. We don't have anything else. What's going on in that place? So we went out. Actually, we failed. The next time we went out, I actually set a bunch of minnow traps and we didn't get it. We didn't catch any. So we missed our window there. We were kind of busy at a short window. Went back in 2003. Got another group. Set it on up, Canada. And I often wondered, just to take a side story here, when I was over at the Children's Museum, I often wondered, probably couldn't get away with this today. Every spring I would go down to the post office in West Hartford, and I would have these boxes with holes punched in them. We would put them in the live animals, because they clip the tails, that's how we do the genetics. We would put the live animals in Tupperware, and we'd moisten paper towels on the bottom, and we would put 1 in each little Tupperware thing, and I would pack them in boxes, punch holes in the Tupperware, pack them in boxes, with some batting and stuff like that so it wouldn't shake around, take the thing shut, put them in a box, regular box, punch holes in the box, label the box, and say live animals, be careful. I would go to the West Hartford post office. I was going several times a week with these boxes, mailing them to Canada. I'm going into the post office, and I never once was asked, what the heck are you doing? Nowadays, I probably have Homeland Security. What the heck is that doing? So, a little difference now. So in 2003, we got more, set them up. Guess what happens? Same thing. Get the call, it's more. Hey, they're all LJs. They're all females. What's going on with that population? So what we're trying to find is who's the parent. And our feeling is that because we know the way they breed, remember we said the females have to have males. They have to have a parent. The parent would not be an LJ, it would be an LL or a JJ, pure. Because we don't have hybrid parents. In fact, when we found hybrids that were males, they're generally sterile. So we're thinking, what's happening here? What is going on? There has to be a male somewhere. We haven't found any male LJs. Could it be that there really are male LJs that can reproduce? Hasn't been seen before. What else could this account for? Why else might we see that? Anything else that could be happening here? Any thoughts? What do you think?
What about that cloning thing that you were talking about?
What about that cloning thing?
Yeah.
Maybe this is an example of a group that's able to clone. Because by the way, as we're doing this, in a place in Canada, they've got 175 individuals already that are all this and all female. So they're thinking the same thing up there. And there's so we're going, what the heck is going on? So we're looking, is it cloning? Is it that there's males that are LJs that we're just missing that can reproduce? Is there something else that could be happening? Now by this time we've collected about 20 individuals. What else could be happening? Any other reason why we haven't figured out who the parents are yet by 2003? If they're cloning, we're never going to figure it out, right? Because they're cloning. We have to actually get them in the lab and so forth. What if we're just not catching the males? What if we're just not catching the parental species? Maybe we're missing them. Maybe they're there all along and we're just missing them. We haven't found them yet. Now remember, they got 175 up there and they still haven't found them. We've got 20-something, we still haven't found them. Okay? So now we go to my call to Christopher and come back and say, geez, we need to go get some more salamanders. Now, you do have to notice that it's a little bit of a gap there. We kind of gave up for a while. Actually, that has to do with funding. It has to do with being busy with other projects and things like that. Because 2003 is the last survey. We did try to get some more in 2004 or 5 and failed. And then I started working at the Connecticut Science Center. We were just building the Connecticut Science Center, which consumed a significant portion of my life, day and night. So I didn't get out to do some sampling. But last spring it was, right? Last spring, which was 2012, we said we've got to, it was on our list. And this was on my list. I have a list of the different areas that I need to get to for different things. With sticky notes, I said, we've got to get back there, Mike, and can you guys sequence some salamanders if I go and collect some salamanders? And he said, yeah, absolutely. Get it, see if you can get some salamanders. So we scrambled and put together a trip, and we went out and collected some salamanders, and I'm happy to say we solved the mystery. In 2012, we found one individual that was LLJ. All the rest were LJ female. We got one LLJ. So what that tells us right there is the parental species is blue-spotted salamander, not Jefferson. We couldn't figure out. And it tells us that they're relying on male populations of blue-spotted salamanders that are somewhere within that area. That's what they're relying on. The guys in Canada still have not found their parental species. So whether there's something else going on in addition, maybe. I mean, this is an odd number. It took us to get to one LLJ. But we found that one, and we believe that they eventually, based on what we found, we think that they're going to eventually find an LLJ or an LJJ or one of the others that will tell them what that parental species is. There is a question, though, which I'm intrigued by, is when we look at the genetic compositions, especially in Connecticut, there's certain areas where we can predict pretty well now based on what our knowledge is. This site at Wintergreen Woods stands out very differently. So when I look at it, I say, what is different about this site? Why would this be? Why is this group behaving differently? Why is this group up in Canada behaving differently? And when I look at, remember all those dots I showed you throughout New England? So we had samples throughout that whole area of New England. In fact, if I go back, can I go back on this thing? Yeah. If you take a look, these are all sites where salamanders were collected over several series of studies where they were genetically analyzed. So there's a lot of sites you can see where we're collecting animals. Why do we find a couple of sites where we have this genetic, none of these other sites are like that. We can find some LJs, but not a lot of them, small percentage, 8% or something like that. So what's going on? Why would we find a couple of sites that are unique like that? Is there something going on here? So to answer that question, or at least hypothesize about it, because we really don't know the answer, but I don't know the wrong way. Hypothesize about it. We've solved it. So we want to say, why is it important? Let me show you why. When we look at genetics, what does genetics do for us? When we look at these organisms, they've been around for about 5 million years. I told you that we've now, James Bogart actually did a great study on this, looked and said, really was able to determine that the lineage goes back approximately 5 million years to western Kentucky, just based on looking at the salamander distributions. What we know about unisexual organisms is they don't last long. Evolutionarily, it doesn't work as well as sexual organisms. And the reason is, does anybody know why? Why would a unisexual organism, just females reproducing, so you don't have a lot of genetic diversity, why would that not work well? Why would you tend to go extinct and not do well over long periods of time.
Wouldn't it be like the same as, maybe like a colony of Tasmanian devils inbreeding, and the genetic material just deteriorates over time?
Exactly. So in other words, you don't have a lot of diversity. If you don't have a lot of diversity, you build up bad genes, and you have no way of getting rid of them. And it also doesn't allow you to incorporate new genes to basically adapt to change in your environment. So it tends to be not as an effective as a mechanism. It allows you to clone and basically preserve your genes, but basically if you get these buildups and you cannot adapt to new things. So with sexual reproduction, it allows you mechanisms to bring in and be healthier, be able to adapt to different changes, and also mechanisms to get rid of genes that really are not functioning or are detrimental to the population. When we look at the group of salamanders that are involved in this hybridization, we can see specializations that they have. Blue-spotted salamander is very cold tolerant. It occurs way north, up into Canada. It's one of the widest distributed salamanders in the northern part. So it's what we could call it an antifreeze expert. The Jefferson salamander, more warm. It's more tolerant of warm climates, more southern climates. The tiger salamander, it's really a robust salamander built for particular terrains and can withstand certain terrains. The streamside salamander, more adaptable to flowing water and different type of wetland conditions. The smallmouth salamander is very more adaptable to mud and clay environments. So by hybridization, you bring in some of that ability to adapt to change. So why is that important? Climate change, development pressures on the landscape, different things that are happening that force these organisms to change over time. So when we look at changes in the diversity, what are we seeing? What's different about the Wethersfield wintergreen wood site that might be different from the other sites that we're studying? Here's one thing. The first reason I started looking at the site. The reason I was interested in it is because it's a very developed area. This is Wethersfield within the greater Hartford area. It's a very urbanized area. So this is an area where you have a population that is experiencing a lot of human pressure from development. Now you may look at wintergreen woods and say, no, that's a great pristine thing, but if you think about it, these organisms really require larger landscapes over time. Let me give you a good example. Remember the salamanders I showed you, the Swedish salamanders? There was one that I said was fairly widespread, the spotted salamander with the yellow spots, black body. It was fairly widespread in vernal pools and forests here in Connecticut. You can't find it in wintergreen woods. You ever found one there, Chris? Can't find it in wintergreen woods. Doesn't occur there. And one of the reasons I believe it doesn't occur there is there's not enough forest. Not enough forest. But yet, there's lots of blue-spotted salamanders there. They're not uncommon there. I mean, I find them, they're there in pretty good numbers, the hundreds, probably even the thousands. They're there. Why are they able to persist there and the spotted salamander is not? There are wood frogs there, but not a healthy population of wood frogs. There's not a lot of them. So I can already look at the site and say, it's stressed environmentally. And if you were to look at birds, I'll bet you can tell a certain thing, because we can look at different birds that are sensitive to different area sizing and so forth. I tend to look at the salamanders. So I look at it and say, does the genetics have anything to do with that? Is there a reason why the blue-spotted salamanders are doing well? And is there something that's associated with the environment that's driving the genetics, or that genetic combination allows them to persist within these environments? One of the things we've seen as we're studying the Jeffersons and blue-spotted salamanders is the Jefferson salamanders seem to be more sensitive to development and urbanization. They don't do well. They drop out quicker. We tend to see them go extinct or tend to not be in areas that are more developed. The blue-spotted salamander, on the other hand, we tend to find more, I would say, plastic or able to persist in an agricultural landscape as long as there's some forest. Here, in a more urbanized environment. So the genetics may actually be influenced somewhat in the adaptability by landscape change, both natural and also human influence through development patterns. So we're very interested in what these populations tell us and what we see in these populations. And when we see an anomaly like that, it's a little different, I start to look around and say, well, what does this tell us about the landscape and what's the interaction there? I don't know enough about the site in Canada where they have 175 and they're all that. I don't know how developed that is. I'm very curious about that. I didn't learn about that site until actually about 3 or 4 months ago. I'm very interested in why they're not finding any parental species, but I'm very interested in is, what's the condition of your site? How many acres of forest? How big is it? How urbanized is your environment? Because I'm curious to see, is there something related to the site that's, that has an influence potentially on the genetics? So when we look at this, the reason we're interested in these things is they tell us a story. We're basically seeing, right here in Connecticut, you right in Wethersfield, you're seeing evolution happen in your backyards, in essence. Because this is the type of interactions that drives the development of new species over time. Remember I said that these guys are not the same salamander that 5 million years ago led to their development. Not the same, they're related, ancestral, but it wasn't the blue-spotted salamander or that streamside salamander. It was a relative, a close relative of the streamside salamander through some hybridization event that led to this whole complex that we're seeing today. And we're seeing it work itself out evolutionary-wise. We're influencing it potentially, I believe, through development patterns. Because they're now forced to deal with changes in the landscape. If they're sensitive to our development, they're going extinct, their genes are going extinct with them. If they're more adaptable to it, they're persisting, their genes are persisting with them. And they're changing in the combination that we're seeing. So there's a lot more to come on these salamanders. Right here at Wethersfield and Wintergreen Woods and throughout Connecticut, we're in the zone of hybridization. So I often think when people are walking through Wintergreen, I've walked through there so many times. I often think when people are walking through Wintergreen Woods, that's the side of Wintergreen Woods, you should lift up a few logs and see if you don't see one of these beasts. They're completely harmless, by the way. They don't bite or anything like that. They can't hurt you. If you won't see them walking around. I dare say 99.9% of the people who are walking through there, walking their dogs, walking through there, and never know that this is occurring every spring and every summer. This is what's happening, and it's getting ready to happen right now in Wintergreen Woods, in those vernal pools and in those forests. So if you're out there, flip a few logs. Make sure you put them back, but take a look under some of the logs, especially in the coming months, because that's where they'll be. They'll be starting to move under those logs. I found almost all the sample last spring under logs. I found, I think, 8 or 9 in one day in about an hour of searching. I got one in a minnow trap. They're big animals. They're about that big, so you can't miss them. Very pretty animals, nice blue. And they'll sit there. They don't run like a lizard. They'll just kind of sit there if you lift up the log. That's happening. Evolution's happening right in the backyard. So I know there was a lot of depth of content here with genetics, so I apologize for that. But that's part of the story. It is an interesting story about how organisms evolve over time, how we potentially have an influence on that, and some of the mysteries of how we're starting to understand about our biological diversity in the world. Any questions?
I have a question. When you get them in the lab, I mean, how much of a part does DNA? Could you tell the story from the lab, or do you have to? I mean, like when you do get the salamanders, you said when you get them in the lab.
Yeah.
I mean, you couldn't tell who their DNA, who their father and their mother.
Yeah, we take the DNA sample, but when we look at that, that's what we get. Is we get these sequences that we say, okay, this one is, and what all of those animals that we were getting in the lab when we would sequence it, it was all LJ.
Same.
So it could have been a Jefferson salamander as the father,
right,
or a blue spot. We had no way of sort of knowing who the parental species was. We were sort of guessing, or if there was a parental species, that was kind of the mystery was, A, who is it? Is there one? If not, why are they reproducing? How are they reproducing without a male? Because we haven't seen that yet with this group of salamanders. Is it a new mode of reproduction? Are they clonal? I mean, we just didn't know. So we're trying to find the answer to that, just like they are in Canada. They kept sampling saying, what's going on here? Why are we not finding a male? Why are we not finding what's going on with this population? It doesn't fit what we know now. But that doesn't mean it's different, because literally, as I said, we used to think, back in the 1960s, right up till, I'd say 1970, actually I would say 1985, 84 at least, we still considered them separate species. We didn't know. We didn't know enough about genetics, and we didn't have the techniques to really start to investigate this. Now that we do, it's progressed our understanding of what's happening to really start to see, well, wow, this is what it is. You know, we used to think it was this, now we think it's this, now we're finding this. It wasn't until last year, I believe, or the year before, remember that one I showed you, the streamside salamander?
Right.
We had never seen the genetics of that salamander we knew it was in the nuclear genome, but we never saw it in the mitochondrial DNA. We saw tiger salamanders, smallmouth, blue spotted, Jefferson. We never saw which was, think about it, which is the ancestor it's closest to,
Right.
never showed up in the mitochondrial DNA.
Yeah.
We knew it was the ancestor until last year. They found one in, I think it was Ohio, it was either Ohio or Ontario, they found one where it showed up in the mitochondrial DNA. They said, oh, okay, it can contribute to that as well. Didn't know that before. So, you know, the more we